Look closely into someone's eyes. Not the color, not the size. The patterns, the folds, the crypts, the tiny furrows that never repeat. Even identical twins don't share them. So here's the question. If our DNA is the same, why are our eyes still different? Welcome back to my channel, where we unravel life's biggest mysteries written in your DNA. Today, we're diving into one of the most precise biological fingerprints humans have, the iris. What shapes its color? What sculpts its patterns? And why genetics alone can't fully explain its uniqueness? Let's start with what genes do control well. Eye color. The strongest genetic influence comes from a region on chromosome 15 involving the OCA2 gene and a regulatory switch inside HERC2. A single variant, RS1213832, explains up to 70% of the blue-brown color difference in humans. Other genes like MITF and PAX6 guide eye development and pigment cell behavior. This is classic genetics: strong signals, predictable patterns, population-level effects. But color is only the surface. Now we move deeper. Iris patterns, crypts, furrows, contraction rings are not controlled by one dominant gene. Genome-wide studies show associations with genes like SEMA3A in crypt formation, TRAF3-IP1 in furrow contractions, and SLC2.4A4 pigmented rings. Each explains only 1 to 3% of variation. That's the signature of a polygenic trait: many genes, small effects, complex interactions. Interestingly, many of these genes are involved in neurodevelopment. The same pathways that guide neurons also help sculpt the iris. Here's the twist. Even with the same genes, even with the same environment, no two irises are alike. Mathematical analysis of millions of iris images, including identical twins, shows something remarkable. The fine structure of the iris is statistically random. This randomness comes from epigenetic events during development, like stochastic gene expression, cellular timing differences, and microscopic tissue dynamics. Over 200 independent degrees of freedom emerge during iris formation. Genes define the boundaries. Epigenetics fills in the details. This is why iris recognition works so well. Not because eyes are designed to be unique, but because development is not perfectly deterministic. Life embraces controlled chaos. The iris becomes a biological record of genetics, development, and randomness, a signature no genome can fully predict. Your eye color comes from your genes. Your iris pattern comes from something deeper, a collaboration between DNA, development, and chance. And that's the story biology keeps telling us. Genes don't write destiny, they write possibility. What other biological fingerprints should we explore next? See you soon.